Types of optical microscopes. There are several types of optical microscopes, each designed for specific applications and offering different imaging capabilities. Here are some common types. Bright field microscope. This is the most basic type of optical microscope. It uses a bright light source and a simple lens system to observe specimens that are visible because of differences in contrast and shading. Bright field microscopy is widely used in biology, materials science, and other fields. Phase contrast microscope. Phase contrast microscopy enhances the contrast of transparent and colorless specimens by exploiting differences in the refractive index of different parts of the sample. It is particularly useful for observing living cells and tissues without the need for staining or fixation. Dark field microscope. Dark field microscopy illuminates the sample with oblique or off-axis light, causing the specimen to appear bright against a dark background. This technique is useful for observing specimens that are difficult to see using bright field microscopy, such as transparent or translucent samples. Fluorescence microscope. Fluorescence microscopy uses fluorescent dyes or labels to visualize specific structures or molecules within a sample. When illuminated with a specific wavelength of light, the fluorophores emit light of a different wavelength, which can be captured and visualized. Fluorescence microscopy is widely used in cell biology, immunology, and molecular biology. Confocal microscope. Confocal microscopy uses a pinhole aperture to eliminate out-of-focus light, resulting in improved image resolution and optical sectioning capabilities. This technique allows for the reconstruction of three-dimensional images of thick specimens and is widely used in biological and biomedical research. Differential interference contrast (DIC) microscope. DIC microscopy enhances the contrast of transparent specimens by using polarized light and Nomashi prisms to create images with enhanced depth and detail. DIC microscopy is particularly useful for observing living cells and tissues in a non-destructive manner. Polarizing microscope. Polarizing microscopy is used to analyze the optical properties of birefringent materials, such as minerals, crystals, and polymers. It uses polarized light and polarizing filters to examine the interaction of light with the sample and reveal structural and compositional information. Differential phase contrast microscope. This technique uses variations in the phase of light passing through the specimen to produce high-contrast images. Differential phase contrast microscopy is useful for observing unstained living cells and thin biological specimens. These are just a few examples of the many types of optical microscopes available, each offering unique features and advantages for various research and industrial applications.